Thank you very much. I really appreciate the chance to come and talk to everyone, both here and those who are watching at a distance. So as a little background, I'm a medical oncologist up in Boston, although most of my time is spent running a laboratory trying to investigate the biology behind gastric and esophageal cancer, and then trying to think about how we could use the underlying biology to develop more effective therapies. And and a lot of the foundation for what we do is based upon the idea that what makes cancer tick is really the changes in the DNA of the cancer cell. And so when I'm talking about the cancer genome, what we're talking about is the DNA of cancer. And just as a little bit of background, DNA is what's in each of the trillions of cells of our bodies. DNA is a string of letters, A, C, G, and T, and those together form the cookbook, the recipe book for everything that our cells do. It's the recipe for making the proteins that do the work of the cell. And you could imagine that just like if you have a recipe for something in your kitchen and that recipe gets a typographical error, then what you're trying to make won't be right. And it's the same thing in your cells. If the cookbook for your body has errors in it, and so you're making the wrong proteins, those cells are not going to act the right way. And so when we're starting to think about cancer as an alteration in the DNA of the cell and an alteration in that cell's instruction manual or cookbook, it's useful to think about the type of DNA changes that can be there. And one thing that often can be a little bit confusing to people is when people talk about cancer genes, they're often talking about two different but related issues. And that's what I'm referring here to as somatic changes and germline changes. So what I'm going to be talking about primarily is what we think of as these somatic changes. And the idea here is that somatic changes are really how the cancer cell's DNA is different than the DNA of the normal cells of your body. And this is because over time, the cells that become cancer acquire changes or mutations in the DNA. And when we start to think about how we're going to implement targeted therapy for cancer based upon the cancer genome, the vast majority of what we're talking about are these situations where the cancer cell has picked up a mutation or alteration that's not in the normal cells, and that's something we're trying to target. Now, this is contrasted with germline or inherited changes. And so these are when people inherit a variation in their genome from their mom or their dad. And in many cases, these are the gene changes that can influence cancer risk. So some of the famous genes people might know about here would be, for example, BRCA1, which is an inherited variation that could impact your risk of breast cancer. And for this audience, a very pertinent gene would be CDH1, which is an inherited mutation that can impart risk of diffuse gastric cancer. But in these cases, since you inherit that alteration, that's in all the cells of your body, whereas in the first case, the somatic alterations, it's really asking the question, how is the cancer DNA different than the DNA in the rest of the body? So with that background, the question is, why do we care about the cancer genome? And one analogy that I think makes sense and is helpful is imagining the cell in your body kind of like a car. And just like in your car, you have parts of the car whose job is to make the car go. You have parts of the car whose job is to make the car stop. So you have your gas pedal and you have your brakes. And there's all kinds of machinery in your car to make the car go and to make the car stop. And just like that, the cells of our body have the machinery, have proteins that make the cell grow and divide. and make the cells slow down. And a lot of the alterations that happen in cancer essentially are screwing up that machinery. So you could imagine you have genes whose job is to make the cell grow, and you would have mutations that could activate those genes, which would be sort of like having a car where the gas pedal is, caught on the floor. And you have other genes whose job is to be the cell's brakes, and those get inactivated. And so one way to imagine a cancer cell would be like a car where the gas pedal is caught on the floor and someone has cut the line for the brakes. And so that cell is growing and growing, but then you can't slow down. And so the way that this would happen would be by those gene changes that alter that machinery that's normally controlling how the cell would be working. And this is important because this idea of genes changing cancer is increasingly important for guiding therapy. So as I mentioned, tumors will develop in part because you activate oncogenes, the genes that make the cell grow faster. And that what happens is that as the tumor is growing because of these turned on genes, the tumor then essentially becomes dependent upon what that turned on gene is doing to the cell. If your car is going fast because the gas pedal is stuck to the floor, that car is dependent upon that gas pedal being down. It's the same thing if you have a gene that's making the cell go, that the tumor becomes dependent upon that. And increasingly, a lot of the new drugs that are being developed now are actually trying to target these various kinds of genes that can be turned on and can be making cells grow. And what is complicated is that if you have different patients coming in with quote-unquote the same cancer, is those different patients won't necessarily all have the same gene that's making their cancer grow. And then because of that, you could imagine if you have different reasons that your cancer is growing because of different genes that are turned on, that it's not going to be the case that the same therapy is going to be effective for all patients. It's sort of like, if you have a car that is going fast because the gas pedal is stuck to the floor, taking your foot off the accelerator is not going to make that car go any slower. But if you have a car that's going fast because your foot is pushing down the accelerator, taking the foot off the gas pedal is going to help. And so I think the big idea now is that as we could try to pinpoint using genomic technologies what's wrong in each cancer cell, what are the genes that are turned on, it gives us new ideas about what might be appropriate therapies to give to each patient on an individual basis. And then, so what are some of these changes? So one type of change is what's called the mutation. And here is where you actually have the individual letters of the genome getting a typographical error, such as you have an A that gets changed to a G. And these can be changes like this, even though small, can be very important and be an important therapeutic target. For example, there's a class of mutations in lung cancer that are caused by a change like this that are very effectively treated now, treated by a small molecule inhibitor called Lapatinib. You could get changes in the genome that aren't a small typographical error, but a bigger error, such as getting extra copies of a gene. Normally, each of the genes in our body, we have 2 copies, 1 copy from mom, 1 copy from dad. But you could get changes where instead of having 2 copies of a given gene, you have 10, 20, 40 copies. And if you could imagine having a car that has 40 gas pedals, that would be a very different car than a car that just has 1. And the example here that's pertinent to this audience is actually the gene HER2 or ERB2, which is a gene that's commonly amplified, has extra copies in gastric cancer, and patients with that alteration now are treated with a targeted drug that specifically hits that HER2 protein, and that's the same as happens in breast cancer. The other kind of gene change we don't have so much in gastric cancer so far, but these are called translocations, where 2 different genes kind of get fused together to make an abnormal gene or a chimera. And just like the chimera in mythology was an animal that was, I think it was like half horse, half something else. But I think that same thing is true here, where you get a gene that's part of two different genes, and can be very famous, such as in leukemia, where there are genes like this that are very effectively targeted by new therapies, especially in CML. So then just a little bit of the background again. So cancer is a disease largely of the genome. You get those inactivation of the tumor suppressors, activation of the oncogenes, and increasingly understanding what drives cancer, what are those genes, can be a real guide to us to think about what are the new emerging therapies, what are the optimal therapies. And very excitingly, we live in an era where we have incredible new technologies to study the genome to give us insight into understanding what these genes are. And so, as I was saying, technology has been a huge boon to this work that we do. And we're all used to thinking about how much technology has advanced in recent years. And when I think about the computer I had when I started college compared to what I'm carrying around in my pocket right now, where it's probably 1,000 times faster and a whole lot cheaper. And the idea of how fast computers get improved came from this idea called Moore's Law from Gordon Moore, who was the president or CEO of Intel, the chip maker, and said that basically every 18 months computers would get twice as fast. And so if you look at how fast computers have improved, which is the white line, versus how fast our ability to analyze DNA has improved, you see that our ability to look at DNA has sort of far outstripped that, and sort of it's been one of the most amazing developments in science in some time. And if people know about the Human Genome Project, which was an effort to map all of the genes in humans, 10, 15 years ago, that was an effort to map the genome of one person who was scientists around the world working for years for hundreds of millions of dollars to map that out. But now technology is advanced, so doing something like that is, instead of being hundreds of millions of dollars and years of work, now could be a few $1,000s in days or weeks of work. And so that means that's unlocked the ability to then look comprehensively at cancer and say, what are the genes that are altered? And so our lab at Dana-Farber has been leading a number of efforts to try to map out what are these genes that are involved in cancer, and then there's a lot of more exciting work going on. And one of the most exciting projects is called the Cancer Genome Atlas, or TCGA. And this was an initiative that came out of the National Institutes of Health several years ago, because as we had this amazing new technology and we could comprehensively analyze the DNA of cancer cells, because we started to ask the question, we should do this for a lot of cases and really create a systematic map of everything that's wrong in a number of common human cancers. And so as we got started, we're fortunate that gastric cancer was one of those tumors, and that we also then got funding to do esophageal cancer, which is very similar to gastric cancer, but because it's often hard to draw the line between the two of these, we decided to actually make a combined project where we're going to look at these together to really map out. And our goal is to get 1,000 cases. I think we're going to get about 700, but to comprehensively map out what is altered in these cancer samples. And I don't want to get into the weeds here too much, but essentially we've completed our first about 300 cases so far, and our analysis is now submitted for publication. Now gastric cancer is not one disease. There's variation in gastric cancer. There are intestinal types, there are diffuse types. There's differences of gastric cancers that happen near the esophagus versus those lower in the stomach. People argue about whether gastric cancer in our population in the U.S. is different than gastric cancer in Asia, and so forth. And so when we did our project from the TCGA, we set out to ask some of these questions, how gastric cancer is the same and different. And I don't want to get into the details here, but we had about 300 cases with comprehensive data, and then we were able to try to ask the question, can we put gastric cancer into rational groups? And in effect, that's what we did here, where we had this comprehensive data and we were able to find 4 different classes of gastric cancer, which we show here. Again, I don't want to go into the details of this right now. And then we were able to essentially divide these tumors into 4 groups. Some gastric cancers had a expression or a presence of a viral infection called EBV or Epstein-Barr virus. Some gastric cancers had a feature called microsatellite instability. Some sort of had what we called a genomically stable phenotype, and some had a phenotype we called chromosomal instability. But we were able to sort of take what was one group of tumors and put it into some rational groups. And up to now, most clinical trials just put everyone in the same basket, and now we could start to piece out how different groups of tumors might have different underlying biology. And then we could look at sort of what are some of the altered targets, what are some of the candidate therapeutic targets in these different groups. And this is looking at each of the individual tumors that was categorized here, where we're in their 4 different groups. And there's a lot of information here, but the key point is that those big areas in red are where you have a gene that's amplified, and those areas in green are where you have a gene that is mutated. And so, for example, second from the top is the gene ERBB2 or HER2, where indeed we found that 17% of our whole group had ERBB2 alterations. But even beyond that, there were a number of other genes that were either amplified or mutated that are candidate targets and targets that should be considered for further testing. So it gives us some idea about what are some of the other targets that are available that we should be evaluating, and also how different classes of gastric cancers might have different targets that are preferentially present. And so as we think about this, what we could do with understanding the cancer genome is we could try to put patients into better groups, and we could try to make subtypes where patients have particular gene changes, particular targets that are present that might help us develop more effective clinical trials in the future, essentially taking what started off as what we thought as a homogeneous group of patients and actually realizing that we actually had different subgroups present. And so the question is, so genomics can inform possible targets. There are a number of targets that are present across many gastric cancers that are candidates. HER2 is one we're acting on right now, but there are other targets there as well that are worth considering, such as other genes that are similar to HER2 and other related factors that are such as genes that involve the cell cycle. And a number of these different targets or different genes are targeted by drugs that are existing or are in current development right now. So having this information helps us think about how we're going to try to bring some of these therapies into patients and what might be the genomic test that would help us predict who would be the patients that should be tried with certain inhibitors. But what's important to note is that these data from the genome are not telling us definitely that any drug is going to work. It helps point to a hypothesis of something that might work, but these tests by themselves are not proof that something is going to work. With the story I've told so far, it almost makes it seem like treating cancer should be easy. You'd have 3 patients. One you'd find an alteration of gene X, one you'd find an alteration of gene Y, and one you'd find an alteration of gene Z. And so you would just say, okay, if you have a gene X, I'll give you an anti-X drug. If you have a gene Y mutations, I'll give you anti-Y. And if you have gene Z, I'll give you an anti-gene Z, and things would be easy. Unfortunately, even as we could develop better therapies based on our understanding of the genome, we still have to deal with the fact that cancer is a very difficult enemy. And when we give these therapies that are trying to attack the real underlying biology of cancer, we might be thinking we're going to put in a roadblock that's going to stop cancer in its tracks. But we have to realize that what cancer often sees when it sees these therapies is a roadblock, but it sees it has a way to go around that roadblock. And so just being able to target the cancer genome is not going to be a simple cure-all. Cancers are smart, and right now good cancer therapies that are highly beneficial are often only temporarily beneficial. And so in the future, we're going to have to learn how cancers try to work around our therapies, how they become resistant to good therapies, and then how we could develop combinations of therapies that are effective, similar to the world of HIV, where initially we had one good drug, AZT, that was able to slow the HIV virus down, but then it got better and then went back up. And now we give patients combinations of inhibitors that are able to lead to durable long-term efficacy. And so I think we have a lot of work to do to figure out how we're going to develop good combination therapies. And essentially, I think it's going to be sort of like playing chess with cancer. You're going to put one roadblock in cancer's way, and you're going to figure, and the cancer is going to try to work around that alteration. And so you're going to try to think, well, if I do this, the cancer is going to go that way, and so I'm going to put a second piece over there and try to develop a strategy that's going to lead, I don't know if to curing cancer, but at least trying to turn many of these cancers into a chronic illness as opposed to a rapidly lethal illness. And so how do we do this? So like in our lab right now, we're saying, okay, we need to develop better therapies and learn how cancers are going to work around our therapies. So we're building efforts to get fresh samples of tumors from different patients and say we're going to try to investigate how these cancers respond to therapy. And we can't do everything we want to in patients, so we'll say, well, let's take a tumor from a patient and let's implant it into mice. And then we'll use those mice as a tool to do clinical trials. And we can investigate cancers with particular genomic alterations in mice and do clinical trials in these models. And that would give us opportunities to figure out how we could try different combinations, how we could test how the tumors become resistant to therapies, and how we could prioritize the best therapies and to bring forward into clinical trials. So where will we be in the future? I think genomic testing will be routine. I don't know if it's just going to be the tumor or if it's going to be circulating tumor cells or whatnot. And I think we're going to have more effective combination therapies based on these data. And we'll be knowing better how to combine these targeted therapies with other traditional therapies such as chemotherapy as well as immune therapies. So last year when I gave this talk, there was the question, what should I do? Should I get my tumor profiled? So increasingly this is possible. There are a number of companies and academic centers that do genomic testing of tumors. This is not standard of care, and I think it's important to note that if we knew what to do with the results, it would be standard of care. So, so the reason it's not routinely done is because it's still research and we don't entirely know what to do with the information. However, having the data on your tumor may give you ideas about possible clinical trials to consider. And I put a lot of caveats in that statement. So it's reasonable to do, but again, if we knew what to do with the information, we would be doing it for everybody. All patients get HER2 testing because we know what to do with that information, for example. So if people who are interested in getting tumors profiled, there are a number of big academic centers who are doing this. There are also companies that are doing this as well, where you could send samples to the companies, and many insurance companies will cover that, but it's inconsistent. And interpretation is still a challenge, but there could be benefits to this, in terms of considering clinical trials. But again, if we knew what to do with this information, we would be doing it on everybody. So if we think about the big picture right now, I think we're doing a lot of work to characterize what's in the cancer genome. And from that, we're going to figure out what are the key genes that are responsible for these cancers, and then figure out what to do about this, how to, based on these genes, what are the therapeutics to use in different patients. And then we're increasingly going to use that to guide how we take care of patients, and then over time we'll figure out how to make this better, how to develop rational combinations, how tumors become resistant to these therapies. And this is going to require a lot of work by many people in the lab and in the clinic. But I'm hoping over time we're going to make this a whole lot better. And I thank you very much for your time, and I'm happy to answer any questions.
Microphone? Thank you very much. Does anybody have any questions? This was a incredible advancements in genomics, and if anybody has any questions, we do have some questions online, so while you're all thinking of your questions here, I'm going to ask one question here. We have, I am HER2 positive and was on Herceptin, but it affected my heart. What other protocols are there for HER2 positive?
Oh, so that's a good question. So I want to talk generally now as opposed to giving particular advice to any individual patient, but there are different types of drugs that will block HER2, and this is, I think, something we need to evaluate as a field. You know, Herceptin is an antibody drug that binds to the HER2 protein. There are also pills that are small chemicals that interrupt. They don't bind HER2 like the antibody does, but sort of try to block its enzyme function, which are also being evaluated. I think so far from big clinical trials, the data for the antibodies has been better than the data for the small molecule chemical inhibitors. But that said, those other drugs are FDA and approved and available, and it's reasonable to consider that other class of drugs if you can't tolerate Herceptin. And I don't believe, maybe someone here would correct me, I don't believe that these drugs like Lapatinib have the cardiac effects of, is that right? Yeah.
We'll talk about this
yeah.
a little bit later, but
Lapatinib has been looked at in phase 3 trials in first and second line treatment with some signal of benefit, but not a survival benefit, so it's not approved yet. And then there's this probably wouldn't apply to this patient because there's a Herceptin conjugate drug called TDM1, which Herceptin is conjugated to a chemotherapy drug that's in trial development now. Mm-hmm. So there are also some other tyrosine kinase inhibitors. There's a drug called Afatinib, which is approved for lung cancer, which is being looked at in phase 2. So Afatinib and Lapatinib are commercially available, but they're not approved for esophageal gastric cancer, and so it's hard to advocate for using drugs outside of a study.
Yeah, I think that's right. I think we should, I think that would be a reasonable idea would be, because there are clinical trials that are evaluating these drugs, and so one way to do that would be to do it as part of a clinical trial. Yeah.
We have a question in the audience here, up front. Okay, go ahead.
Could you comment on Dr. Watson of Watson-Crick fame's creating a little controversy about the genomic approach?
Was this his comment about, which comment was this about? This wasn't the one about curing cancer in 2 years, or this is more something he said more, if you could remind me about what he said, that would help.
I read it yesterday and I can't.
Oh, it was something from yesterday.
Well, no, it wasn't.
Okay.
it could have been old.
Yeah.
I was Googling around and read it yesterday. And I don't know when he made these comments. I think it's within the last year. I don't know.
Did he make comments that were critical of the?
Well, I think he was creating a little controversy about the fact that the results are disappointing and that we might be on the wrong approach with the genomic approach. And I was wondering if anybody knew anything about what he's trying to say.
So I would say, I think looking at the big picture, I think it might be a question of people's expectations. So for example, I showed this slide earlier about that Time magazine or Newsweek magazine cover saying this is the war on cancer, these are the new bullets. So that was from a good 10 or 12 years ago, where there was a lot of excitement because there was this drug, imatinib or Gleevec, that was highly, highly effective for a type of leukemia that had a particular gene change, a particular translocation. And in that case, for this kind of leukemia, simply taking a pill that blocked that one key gene was immensely effective and transformed therapy of this cancer. And I think there was some hope at that point that we would be able to turn a number of other cancers into that situation that you could find the one key gene and give a pill that turns that off, and you would have a miraculous recovery for the tumor. And I think it's definitely the case that that model is the exception rather than the rule. And I think then you could draw the analogy to HIV, where we did have one good drug initially, AZT, which was able to lead to dramatic but very transient benefits, but the virus could work around that good therapy. And I think we're seeing that same phenomenon now in other cancers, that any one good targeted therapy by itself is not going to be effective. And I think we might have set our expectations too high, thinking we would just find the one right drug for the patient, and that would be enough. But I think rather we have to think about, as I was saying, this game of chess, where we understand how the tumors are work around our therapies and develop rational combinations. That said, I think there's not, it's not only going to be these new genome-targeted drugs. I think, some people might have thought we were going to use these and we weren't going to give chemotherapy anymore. I think that's not the case. So there's also questions of how to effectively combine these things with chemotherapy. And the other area of a lot of excitement I didn't talk about is a bunch of the new immune therapies that are coming out that have a lot of excitement now. And so I think ultimately we have to figure out how to bring all of these different therapies together, and also how to, when you're in a world where you have so many different drugs against so many different targets, and every week the pharma's coming up with a new inhibitor to this or that, how do you figure out who to give what? And in the past, the question was, what's the best drug to give to everyone with gastric cancer who walks in the door? And we realized that's the kind of approach that's going to give us benefits of 1 month at a time. But I think now we can figure out what's going on for each patient's tumor and start to think about how to bring together these different modalities and how the tumors are going to work around that. So I think he's right that it's not going to be the simple one drug for one patient, but I think that this approach, combined with other approaches, can lead to long-term benefit, but it's going to take some time to figure it out.
Great, thank you. We have time for one more question.
I'm Ann Fonfa, patient advocate. I'm wondering about, my advocacy is in terms of natural therapies, and I know that exercise, stress reduction, and nutrition can affect our genes. Can you comment on that a little bit?
Oh.
I think we can answer the comment. Kathleen was.
I'm sorry?
Kathleen was here as well.
Okay. I could also defer to some other opinions on this, but I'd say there are changes to genes beyond just what I talked about. There's what's called the epigenome, which is how the genes are sort of regulated, turned on and off, and definitely these other phenomenon can have effects upon that. Probably less so upon the actual mutations that are present. But I think that I think everything like that can become intertwined in the context that these mutations happen in, can be altered by the other environment of the cell. And I don't know if you want to comment more upon this.
Okay, so if you're looking at the complementary therapies of nutrition, fitness, and mind-body, yes, they do have an effect. But if you are looking at this as your primary treatment for cancer, that would be exceedingly foolish and unrealistic. You want to look at these therapies as an adjunct to traditional, whether it would be the chemotherapy, the immunomodulation, that type of thing. To use the complementary therapies as sole treatment, it would not be wise. And if you look at parts of the world that only have access to herbs or to the acupuncture, these types of things, and you look at their survival rates, they are far different than the parts of those countries such as China, where they do have access to modern Western medicine. So while yes, you do have some changes, it's not sufficient or adequate in order to that be your primary treatment for your cancer.
Okay, great. Thank you. We're running out of time here. Thank you very much for a great presentation. Next, I'd like to introduce to you